and find, right? Inputs and define the functional foods. And the other one, you can follow Professor Rampin because he starts to and he's going to lead to the other room that will help the seniors for theme number 3 and 4. Right? The diagnosis and the machine. Yes, after the seminar, we will have the sightseeing around the university and until maybe 10 minutes before noon. And after that, we're going to lunch. Okay, so we'll have the seminar here. And I told Professor Anteri to be strict in the time because we have to finish until 5 and 10:40 to start the sightseeing. I think the bus will take us right to the back. 10:40, right? Okay. So our first speaker here. Okay. So Professor Henry, can you guide the persons to the other room?
Ele não vai sair aqui.
Vai sair só lá. Can we start? Okay. Just a minute. Pode ser. Às vezes daí, se alguém precisa conversar, fica mais tranquilo aí. Não, se conversar lá fora, pode ser. Ou se quiser deixar uma fechada, uma aberta, já bloqueia um pouco. Ok, so let's start. I will have to use the microphone because of broadcasting. So, good morning, everybody. On behalf of UNICENTRO, I would like to welcome you. I would speak some words before technical issues about our department, agronomy department. We are here in Unicentro since 1999, but our first class was graduated in 2003. We have started here when our streets weren't finished yet. We have this kind of vision, but today we have better infrastructure for teaching and also for research. Some labs, maybe Professor Jackson said that maybe we can visit if some of you have interest in going to our department. We started here, most of our professors in 2003, those who are present here, some are of 1999. And in 2007, we started our postgrad program. And last year, we reached 17 years of running. It may seem few years, but it has brought us lots of work. Sorry, the projector is not perfect. But we are 21 professors in this program. All of us, we are focused on this plant production, as we can say in English, area. We take two research lines. One is crop science. So we work with production systems and management practices, mainly for these crops: soybeans, corn, bean, barley, wheat, and pasture crops. But for corn, we have one professor that works with plant breeding. So the second research line is called for us horticulture, and the same kind of studies, but applied to vegetables, fruits mainly, but also to medicinal and aromatic plants. Since 2007, the master course has already finished 274 theses. We have now 18 grants for the students. And the doctorate started in 2014, have already finished 56 theses. We have for them 28 grants. The proposal today is to talk a little bit about yerba mate, or in Portuguese, erva mate. Ilex paraguariensis. I have had the help of Tainá, which is here, for some data, some photographs, and Professor João is here with us. He is the real specialist in erva mate. He has brought us some seedlings, and he had the help, the collaboration of Naldo for some data that I will show you. In this figure, we have the area occupied by our native Tupi-Guarani people and the area of natural occurrence of yerba mate. In red, we have the Jesuits villages. So, Brazil, in these states, Mato Grosso do Sul, Paraná, Santa Catarina, Rio Grande do Sul, 4 states from Brazil, Argentina and Paraguay are the natural occurrence area for erva mate. These people was here already having different uses for this plant for centuries before the Portuguese and the Spanish. Many of the Jesuits here were Spanish. The Tordesillas Strait had this line, but the Portuguese went a little bit beyond the line. And About a perspective about the production, 953,000 tons or million kilograms of yerba mate in Brazil, 812 in Argentina, and smaller production in Paraguay. From this production in Brazil, we have to know that it comes from plantations made by men, and this is called in Portuguese a pleno sol, under the sun, which give us a different result from native underforest, natural curing yerba mate. In this kind of production, Paraná is responsible for 87% of total production of native underforest yerba mate. But together with plantations, Paraná has 63% of total production of yerba mate in our country. This brings us 1.2 billion reais per year. This was for 2022. In dollars, maybe we divide by 200 million dollars, which is considerable for us. This is the aspect of the plantations, full sun. The plant may be conducted as a shrub, but it may also grow to have bigger sizes. And this is the natural shaded condition. This is our symbol pine plant. These branches of this plant are in our flag from Paraná State. And this is a natural reminiscent of our forest, Ombrófila mista, from Paraná. But for what we use erva mate? Erva mate is used to produce mate, which is mainly the leaves, but also green branches. And it's finely grounded to produce this powder, and it is used with hot water for an infusion. This is the ancient way to consume erva mate. In Paraguay, it's more used a coarse grounded yerba with more presence of branches, and it's for refreshing in hot days with cold water, icy water. It's very common that we have in every supermarket and many other places the tea bags with the yerba. We have some, I cannot say that it's new, but it has been growing, the preparation of industrialized beverages with yerba mate. So this is an example of a new enterprise in a neighbor city. This is João. This is from United States. This Guayaquil is working here, but the capital of this enterprise is from the United States, and they are exporting for their market as an organic appeal, with an organic appeal. As erva is present in our culture, so many other things are starting to be prepared. Cosmetic products is one of these new uses. But the aluminum or plastic capsules to use with Nespresso machines is also a possibility. So this is starting to grow, and the erva is going to different markets. The research I will talk about is the production of seedlings. This is our focus from now on. In this natural environment, the conditions, the natural conditions to the plant, to erva mate, is fresh to cool, humid climate. So here we are in a place that we have more or less 2,000 millimeters of rain per year. And in the hottest month, we have under 22 Celsius degrees. We have frosts in winter in this place. But for us, what is important is that the plant lives with subtropical soil conditions. These soils are called in American classification oxysols, international classification latosols, and they are known to be physically good soils, low density, high porosity soils, but very leached soils because of the volume of rain. So it results that we have soils that are poor in basic cations, poor in available phosphorus, low pH, very acid soils, and they have high potential acidity. This is the condition for the plants, but it lives perfectly. It thrives this environment. So how is it possible? The plant is naturally tolerant to acidic soil conditions. It is also known as an aluminum accumulator plant. Although this aluminum is not available for human absorption, but this is a feature of this plant. In this condition, it has to be efficient on phosphorus absorption, because if you have aluminum in the soil, you don't have free phosphorus for plant absorption. And to start new plantations or to enhance the population of plants in natural conditions, we have to have good seedlings. And in nursery conditions, the substrates and the use of nutritive solutions with low aluminum levels, with slightly acidic to neutral pH conditions, and high calcium, magnesium, phosphorus, and potassium levels is something very different from the natural conditions for the plant. And this raises a question: does fertile substrates and fertirrigation play a role on the rooting of yerba mate seedlings? Because nurseries have problems with rooting, poor rooting for some, for part of the seedlings that are produced. And maybe the high fertility is not a good condition for this stage of the crop. I will talk about the methodology. We have performed an experiment in a private nursery together with Tainá. Here in Guarapuava, we have available a clonal garden. In fact, many of the seedlings are yet produced with seeds, which give us a variability, a genetic variability, which is not so positive. But here we had a clonal garden of the cultivar BRS BLD Alpaba. BRS is used for all Embrapa cultivars. Embrapa is our national agriculture research enterprise. Then we had these cuttings, and these cuttings were taken to plastic tubes with the substrate that is used by many of the nurseries. The Carolina soil is a standard for these enterprises. It has Canadian peat, vermiculite, organic waste, and limestone. So again, with limestone in its composition, it will be a different environment for the root in relation to natural soil conditions. We tested 5 rates of aluminum in the substrates: 0, 0.36, the double, and the double again. And we used as our aluminum source, aluminum sulfate, which is easy to find. We have performed this experiment in randomized block design with six replicates and 20 seedlings per tray. This is the example of the tray, the plastic tubes, and the seedlings. And 20 plants were used, and some of the plants were just boundaries. From this stage on, we have used the nursery standards, so irrigation and irrigation were performed as the nursery normally does. The seedlings were evaluated in 90 days after transplanting, 150, 215, and 300 days after this, the start. I will have to use selected results to show you, so I will show you here in methodology what we have evaluated with score scales to evaluate cloud consistency. This is poor cloud consistency, and this is considered good cloud consistency. Poor rooting leads to losing the the seedling when the farmer is in the field. So this is what the nursery wants. And this means problems, because when farms lose lots of seedlings, they will come to the nursery, and the nursery have to give him more or have to give him back his money. So this research was focused on solving a problem of every day in the field. We also performed a Dixon Quality Index evaluation. The index uses the total dry matter of the seedling, uses the height of the seedling, the neck diameter of the shoot, the aerial part dry matter of the seedling, and the root dry matter of the seedling. So this is a normal index that we evaluate for seedlings, and the Dixon quality uses some other additional evaluations. Oh, we could talk about nutrient absorption and the results in the substrate, but I will have to show you just some few results. Here is the result for leaf area index. When we look to 90 days after transplanting his under, and 300 days after planting. So close to 1 year. Né? Professor João has brought us some seedlings. Those seedlings these are 2 years old, and those are 1 year. I don't know, maybe we can pass some seedlings for them to see. So it is very vigorous, but this is already 2 years old. And for selling and for the farmers to plant, they prefer that kind of seedling. So 1 year, João, more or less? Yeah. 6 months. 6 months. But it's 2 años. 6 meses. So here we have 300 days. More time than this, but maybe here it has escalated a little bit. We can see that already at 90 days after transplanting, or almost 1 year, the rate of 9 grams per cubic diameter of the source, aluminum sulfate in the substrate, has given a better result than not having aluminum. So the plant responded positively to aluminum addition in the substrate. This is the result for cloud consistency, which is those scores we have used. We also have the same pattern, close to 9 grams per cubic diameter is a good rate for aluminum. So in this case, in this photograph, we have called the control 0 aluminum T1, treatment 1, and then the 9 is here. Yes, 9, the fourth treatment. So we have this kind of image when we have good root development and cloud consistency. Yes. Here is the Dixon quality index. It has brought us the same behavior of the plants. Close to 9 grams of aluminum sulfate has given us a better quality of the seedlings. All those attributes, plant height, neck diameter, and the relation between aerial part dry matter and root dry matter indicates that this is the best tested rate for this experiment. We have performed another experiment, and we used not only aluminum sulfate, but calcium, sources of calcium, to know if, aluminum and calcium, they have opposite roles in cell walls building, and we were curious to know if the plant would respond to calcium. But today, just to talk about aluminum, we have given the nursery a response that they should have aluminum in their substrates. The conclusion for this short presentation is that yerba mate seedlings are positively affected by aluminum presence in the substrate. For this study, 9 grams of aluminum sulfate was the best studied rate in terms of leaf area, cloud consistency, and Dixon quality index. I would like to thank your attention and thank our partners. Tainá is here, thank you. Our federal government grant responsible. The working conditions in research by our program, postgraduate program. And I will leave our mail here. Professor John is also here with us. We can talk a little bit more if you have interest. Thank you.
Go to the other presentation, or if you want, we can do a lead some discussion here right now about this topic. Okay.
How many varieties you have with the mate?
More than 30.
30 varieties, selection.
Erva mate is a crop that is studied by the Embrapa, which is federal and have many centers across Brazil. But here in Paraná, we have the forest Embrapa. And this is the crop is studied by this center. Embrapa alone has 30 varieties of erva mate, but we have private organizations of the nurseries in Rio Grande do Sul and other places. They have their selections on occurring materials.
Essa cultivar foi selecionada para produção, não para propriedades químicas. Eu acho que vale a pena lembrar. E existem outras com propriedades químicas específicas.
Tainá is remembering us that there are different objectives on selecting the plants. Maybe the volume of the sensitivity is something important, but maybe one is looking for a particular chemical or biochemical characteristics for this plant. For exportation, they need one feature, and maybe for Brazilian consumers, we have another profile. As in Paraguay, for tereré is different, the infusion is different from Brazil. In Brazil, we consume the fine grounded powder, and it's not so bitter under the shade. But in Argentina, in Uruguay, they prefer a bitter one, and they use more the branches, and it's very different. But there are different styles.
Essa é considerada uma cultivar doce ou com menos teor de cafeína, essa alpada, essa específica.
This one has a smaller level of caffeine. So it's a stimulant beverage.
Can I? So I'd like to thank you very much for your presentation. You show us the five stage of the kind of the seedling, and that's the poor seedling is caused by the nutrient, for example, the AG3 plus or some disease or some the plant condition, what do you say that when we planted the plant is not good. So how can you see that's the difference? And if you explain some, please.
Why we used aluminum? Okay. I believe I'm not a specialist in the plant, but for this master thesis, I had to study, and then I will have to say that it's my opinion. Maybe Professor João has another opinion. I have spent some time in Germany studying aluminum and corn plants. There are plants that are resistant to aluminum, and they leave aluminum outside the plant, and there are plants that tolerate aluminum inside. This is a plant that tolerates aluminum, but as it naturally occurs in a soil that is very acidic and rich in aluminum, the plant has to be adapted, or resistant, or it has to be tolerant. As in an aluminum soil, aluminum-rich soil, there is no phosphorus available to the plants, and in fact, these latosols, they lose basic cations, and then the soil gets acidic. Then it's a whole package of natural conditions. If the plant lives well, thrives in this environment, it is efficient on the use of nutrients. It gets these nutrients from the soil that other plants cannot get. So it has affinity, I would like to say. The Michaelis menten is very low for this, all the nutrients for these plants. So when the plant finds aluminum, a adaptation process that it's capable of doing starts. So is there aluminum, low levels of nutrients, a part of its genome starts to run a physiologic response and creates the adaptation process. If you have high levels of nutrients and you don't have aluminum, this process of adaptation does not start. In fact, as we could say for soybeans, if there is a lot of nitrogen in the soil, the node genes of soybean will not be activated. it will not invite the bacteria for biological nitrogen fixation to get in. In fact, if there is a lot of nitrogen, the soybean plant will resist to the entrance of this bacterium. So it's the same kind of, for me, it's the same kind of thinking. If the environment is natural, it starts to evolve, and if there is a low level of nutrients, what should be the response of the plant? To have more roots, to reach more nutrients that are away from the tree, to explore a bigger volume of soil to get some nutrient. And this is what makes sense for me.
Okay. I think we will have two more questions, okay? Because of our time. So, Paula, maybe you can ask in Portuguese and Professor Marcelo translate to everybody, okay?
É porque é importante também que a qualidade das folhas ela seja influenciada pelos três tipos de produção: a nativa, sobre sombreamento e sobre o sol. E além disso, nós temos também o processo que é muito importante. The process of the leaves to make the tea is very important for the finished product.
In fact, we have some researchers. Yes, she was saying, she was making a compliment that the way we produce leads to different quality of the leaves, and the way we harvest and we treat after harvest will also have a role in the final quality. I believe this would be better if Professor João could explain us, but we don't want oxidation. So the smaller time possible between harvest and initial drying is very important. So we have to dry, to stop any oxidation process, and then we'll have a second time, that is to prepare. We say in Portuguese, cancheada. Então, it's not grounded by metal equipment in industries. It's like a traditional way to do in the field with wooden equipment. So it's traditionally it has a appreciated result here in Brazil.
Small or biggest industry, they have different kind of
Yes.
making the process.
Yes.
TISA, the University of
Federal University.
Yes, Federal University of Paraná. We have a TISA about this, the processor and the different kinds of production.
In fact, here I'm just talking about a small part of a specific issue, rooting for seedlings. But here in Unicentro and in other universities, there are many other studies. Professor Sueli Kinaya from chemistry department studies the availability of metals that are accumulated by the plant in the infusion for human consumption. If you go here to the Engenharia de Alimentos, food engineering department, they study the presence of yerba in the preparation of breads and lots of foods. And we could have many more professors here talking about many different things, but it's a cultural crop for Paraná, for Argentina, and also for Paraguay.
The last question, okay? Just because.
Anyway, I'm very interested just focusing the one of the Yebara Mate. This is a nice approach to enhance or develop new industry finally, I think. Then, yeah, initially when we planting something on the ground, maybe we are thinking how farmer can easily grow it or prohibit some disease. This is the most important point of selection of some variety that you are talking about the taste, potability sometimes. Bitter taste is not good, right? This kind of approach is the second step, I hope. Then you can, yes, provide a new type of mate tea or then finally, because I'm a food scientist, recommend you some biological important things in mate. If you can choose such final step of healthy concerning production, we are very happy to. Then another my comment is about how to grow the root. In my colleague are putting some polysaccharide on the ground. The growth of the root is very, very good. It's just my comment. If it is a good approach, I'm very happy also. Thank you very much.
I don't know if I understand it all, but we could use another other inputs to make the root growth. And you know someone that is testing polysaccharides for root growth? Crab, yes, crab. Okay, yes. As I am from soils and plant nutrition, I have used aluminum, but I believe that there are many root promoters that may be used. In fact, the only different thing we tested in this experiment inside a private nursery was to put some grams of aluminum in the substrate. But Tainá is there. They use as a standard a root promoter as we have the cutting. So we have also used the cuttings for every single seedling. So the control without aluminum and the aluminum rates received a starter as soon as we cut in the clonal garden, we cut the plants and we had the cuttings. It was treated with promotor de raiz. Aya, o promotor de raiz que a gente usou inicialmente
era uma base de boro, que era a base de boro e cobalto.
There is a product that they use, but rooting of cuttings of yerba mate is very difficult, and the survival of the seedlings from having the cuttings or with seeds is not very good. Maybe 50% of survival from start. If I have 1,000 plastic tubes with cuttings, in the end, 1 year later or maybe 6 months, we will have 50% of that. So rooting is something difficult. But if the seedling survives, we found that a natural environment thing that is aluminum is already enough to promote rooting and to have more survival of the seedlings. But maybe Professor Cacilda is not here. She studies growth promoters as microorganisms. Organisms. There are many fungi that are being studied here, and maybe some biochemical compost may also play a role. Maybe testing all together we will have a better result.
Okay, there are a lot of many other things I think to discuss. Maybe we can do some joint research in this area. with working, researching erva mate. There are a huge production here in Paraná, as you saw in the pictures, in the slides. So maybe we can do something in this area. Well, I would like to invite Mrs. Isidoro and Mrs. Kutz to talk about bioinputs, the Green Revolution Transforming Agriculture. They are both PhD students. They are both under my supervision. And and also, I have to just to remember that there is coffee there and some snacks outside. So if you want, feel free just to take there. Okay. Well, so you have until between 10 and 15 minutes to the presentation, right? Okay.
Good morning, everyone. My name is Simone Isidoro Morimitsu, and I am a PhD student at here, UNICENTRO, in plant production. Okay. Today, we explain you about bioinputs, the Green Revolution transforming the agriculture. I am in this study. We evaluate to how the trichodermas fungal transforming this sustainable agriculture. So this study is founded or supported by CNPq, okay, and carry out at UNICENTRO and FAPA. FAPA is a corporation of the, at here in Paraná. Não está indo. Aí. So agriculture is influenced by various factors such as climate crisis, pandemics, geopolitical conflicts, and many others. And these challenges and these factors influenced by food production around the world. These challenges and specifically the climate change promote new plant disease and others changes in the agriculture. However, these challenges also bring opportunities. One's opportunities is bioinputs. Bioinputs are products, services, or technologies which we have used biological origin products. I have a text by support. Bioinputs are improving soil health and enhancing resilience and contribution to one health. Okay, in this part, our research began in 2001, and we prospected the new isolates of trichoderma fungals in a soil of mixed forest umbrophilus, ou floresta de umbrófila mista, né? In this case, we collected of soil samples, selection. We promoted the selection of the best inhibitors of Coletótricum lindemontianum in my case, and Sclerotinia sclerotiorium in the research of the Cynthia. And then we identify of this trichodermas isolates. After that, we had laboratory experiments at here, UNICENTRO, applied microbiology laboratory. And we evaluate the efficiency of these isolates of trichodermas fungal in controlling these pathogens and to this disease of the plants of soybean and common bean. After that, we had confirmation of the efficiency of trichoderma isolates in disease, this anthracnose, and white mold in soybean. Sorry, I had nervous. After that, we've had field experiments, and today now we doing thesis writing, and now Cynthia we'll talk about the results.
Good morning, everyone. My name is Cynthia Kutz de Matos, and I am agronomist and a PhD candidate. And I will show you some few results of our both studies. After the collection of the soil samples in the rhizosphere of Araucaria angustifolia, we obtained 32 isolates of trichoderma, and initially we identified the trichodermas based on their characteristics, morpho and micro and macro morphological characteristics of their colonies. And after we carried out a molecular identification of the isolates at UNICENTRO Plant Molecular Genetics Lab. And after the molecular identification, we identified isolates belonging to species Harzianum, viride, viriens, Atroviride, Coningopsis, and others. These isolates were tested for the two diseases that we are studying. So for the work that Simone is doing, we tested for, she tested for Colitoticum lindemuntianum. So the trichoderma isolates have potential to control Colitoticum lindemuntianum in vitro, and some antagonistic mechanisms show it is mycoparasitism, antibiosis, competition, volatile organic compounds, and non-volatile organic compounds. Simone has made this work with leaves of common bean, and she evaluated the area under the disease progress curve, and the isolates and the control treatment had the highest disease progression. The most promising isolates are the Trichoderma azevedoi, Trichoderma coningopsis, Trichoderma asperellum, that outperform not only untreated control, but also the chemical treatment. And the mechanisms observed for the control to Sclerotinia sclerotiorум, we saw mycoparasitism, volatile organic compounds, and non-volatile organic compounds, and we can see to Sclerotia mortality using the sparse solutions of trichoderma. Regarding to the results of trichoderma potential to control Sclerotinia sclerotiorум, we observed the mechanism, the principal mechanism observed is mycoparasitism. The inhibition rates of the pathogens by the trichoderma isolates in these studies range from 44 to 83%. And the highest high inhibition rate was observed with trichoderma variants. And this result is important too, because the isolated trichoderma variants showed the highest mycelial growth inhibition rate of the pathogen with 50% of decrease. So the future perspectives of our work. We finished the initial steps, isolation and identification, efficacy assessment, and field trials. And now to move forward, we are going to do the formulation, regulation, large-scale production, marketing, distribution, technical assistance, and support. But to achieve this, we need to, we are seeking partnerships to support and funding this project. We received a great support of our advisors, PhD Adriana Kenobi, researcher in applied microbiology, PhD Cacilda Rios Faria, researcher in phytopathology, in PhD Jackson Kawakami, researcher in major crops, and PhD Paulo Roberto da Silva, research in plant genetics and biotechnology. Thank you for your attention. Feel free to contact us if you have any questions.
We were looking if the bus can pass until to do the sightseeing. Okay, 10:40. But thank you very much to keep the time. And I don't know if somebody has some questions. We can do it later. And we were doing with the mission in other University and UEN and Maringá. All three universities have this kind of similar research, and so very important. And okay, so thank you very much. Well, now I invite the next speaker is Mr. Brojan. They were both PhD student, but Ciro he's in the undergraduate student. I think he's the third year undergraduate student. He's going to talk about aeroponics, and we are using some nanoparticles and microparticles with the aid of Professor Naj. He's there, and we are working together in this team. Also, we had some many similar topics in UEL and UN also, these nanoparticles and mini particles. I think here, what's the difference that we are using it to produce potato seed, mini tuber for the basic potato seed. Okay, Ciro. So please, you have 15 minutes, and we finish with our last presentation. Okay.
So, good morning, everyone. My name is Ciro Brojan. I am an agronomy academic here in UNICENTRO. I am in my third year of my graduation. My advisor is Dr. Jackson Kawakami, and today I will talk about the use of microparticles in potato seeds cultivation in the aeroponic system. Well, talking about the potato, Solanum tuberosum L, it's originally from ants in South America. And today the potato is the third vegetable source of food. And looking this high examined, I bring here two examples, two countries with their respective productions. We have Brazil with a production of 4 million tons per year. And it's also important to talk about our state here in Paraná. We produce almost 20% of the total production of Brazil. And the Japan, we have a production of 2 million tons per year. Well, looking this examined and the importance of have productive crops, productive potato crops, we need high quality seed. And talking about the seed, it's important to see that we have the seed, it's the highest cost of the total cost of potato crop. It's almost 30% of the total cost. Well, talking about another way of the seed production, it's the aeroponic system. And basically, we have models, as we can see here, with where we have in the part of the model the leaves, the stems, and inside the model, we have here suspended roots, stolons, and tubers. And we can see here we have the sprinkling system. And the sprinkling system gives to the plant nutrients and water. And it's also important to talk about this kind of production. We produce G0 seed that we call mini tuber. And the mini tuber is free from diseases and the generical problems. Well, looking these conditions, we did the project 23-24, that we produce seeds, potato seeds, inside the greenhouse and using the aeroponic system. And we use it in different treatment, the microparticles that I will talk now. But before talking about microparticles, it's important to talk about the collaboration of two research groups of UNICENTRO here. Our group, the Research Center in Large Cultures of Dr. Jackson Kawakami, and we work with agriculture and basically the mini tubers. And the nano and the applied nanostructured system laboratory of Dr. Nage Sarkali. And he produced for us our microparticles that we use it on our project. Well, but what is microparticles? Basically, are particles with dimensions from 1 to 100 micrometers. And this little size, it's important when we think about the higher absorption potential and a bigger contact surface. And inside agriculture, we see the influence on the plant physiology. And it's also important to talk about the origin. We have two different origins, the inorganic and organic origins. Talking about the inorganic origin, we have metallic microparticles that we see here, silver and copper, that helps on diseases management, and we also see boron, iron, and zinc that helps on the plant nutrition. And we have other different inorganic microparticles that are the metallic oxides. Basically, we have zinc oxide and titanium dioxide. Well, talking about the organic origin, it's basically biodegradable materials. As we can see, the carbon, where we have the cellulose, chitosan, curcumin, and we have to the microherbicides, where we have proteins, polysaccharides, polymers, and lipids. And I will talk about the microparticles that we used on our project. We used curcumin that helps on the physiological modulator through hormonal influences, and we used also curcumin plus chitosan that have an antimicrobial effect and antioxidant activity. And the last microparticle that we used was chitosan that have an active ingredient library, and it's important when we think about the plant organ regeneration. Well, this is the greenhouse that we used on our project. It's here in Campus EDTEC. And I will talk about the aeroponic models that we used on our project. And basically, we have a model with wooden base. We used polystyrene panels for the walls, and inside the model, we used black polyethylene. And when I said before about the sprinkling system, it's important to talk about the empty cone type nozzles that we used. And we used also submersible pumps working with a pressure of 50 psi. We can see here the module and the real module that we used. Well, talking about the experimental design, we used randomized block design plus split plot. We did 3 replications and 8 subplots, and in total we had 24 plots. We used also 4 rows, 8 columns, and a spacement between plants of 12 centimeters, or better, 0.12 meters. We can see here the subdivided plot for each variety that I will talk now about the varieties that we used. We used Agatha and Atlantic. Here we have another image of the inside of the greenhouse. We can see the models, the plants, and all our project. Well, talking about the treatments, we used nutrient solution, the control, and curcumin plus nutrient solution, curcumin plus chitosan plus nutrient solution, and chitosan plus nutrient solution. As we can see here, all of our treatments. And talking about an important topic here, I will talk about the application. We used sprayers, and basically we had 0.6 ml per spray, and we did 6 sprays per plot. In total, we had 3.6 ml per plot. We did 2 applications in 2 specific days. 20 days after transplanting, when we had the starting of the stolon growth, and 46 days after transplanting, when we had the maximum period of the crop and the tuberization period, right? Talking about the final considerations of the project 2324, we have the microparticles can influence the hormonal activity of potato seeds and plants, have positive interference in the electron transport chain of the photosystem, and it's important, it's interesting to see here that we have positive effects on potato seeds with the effect of the microparticles of our treatment persisting through the advancement of the tuber generations, or better, crop after crop, we have there the effect of the microparticles. It's also important to talk about our evaluation. We did weekly harvests. We harvested mini tubers with a size of 25 millimeters or more. We counted to the number of stolons, the number of tubers, and the fresh weight. And it's important, we finished the project 2324, and we did the sequence that I called here project 2425, where basically we did field planting of the G0 mini tuber seeds of the project 2324 of these two varieties that I say before, Agatha and Atlantic. We used control curcumin, microcurcumin, curcumin plus chitosan, and chitosan. This was our 4 treatments. And it's also very important to talk that we didn't use fertilizer in this experiment because we didn't want to mask the effects of the treatments. And we don't have more information because we used the farmer's management. Well, I will talk about some important days. We planted on October 3rd, with desiccation on December 27th, the experiment, and the harvest, and we did the harvest on January 10th. And the cycle of the crop was more or less 85 days. Well, talking about the final considerations of the project 2425, the microparticles, chitosan plus curcumin, promote an increase in the fresh matter in type 1 class of potato seeds. And we have more potato seeds in type 1 and 2 classes. But these effects we saw only in the variety on Agatha variety. And I would like to suggest the need for partnership with Japan to study the use of artificial lights to optimize the production and the quality of mini tubers, and this study the optimization of large-scale production of aeroponic model for farmers. We have here the image of the field experiment. We have here the Agatha plant. We can see the tubers, and this, we didn't use fertilizer. It's important to see here, and we have good and excellent effect and excellent result on Agatha variety. So thank you all, thank you everyone for the attention.
Thank you for keeping the time. So we can, I think, finish until the time we are thinking. I don't know if somebody has some questions, some doubts. Professor Watanabe, please.
No es pregunta, comentario. Luego podemos falar respecto a cómo ver la colaboración con ISOs en japonés. Y hablé con el doctor Jackson Kawakami respecto a posibilidades. Okay.
Thank you, Dr. Watanabe. So you should start learn Japanese, right? Some Japanese. Okay, no other questions? No? Okay, thank you. So now I would invite, thank you, Ciro. Thank you very much. Our last speaker, Emanuele, Mrs. Emanuele. She's working with the advisor of Professor Nagy. Professor Nagy is from the pharmacy, right? The pharmacy. Oh. I don't know. Oh, start intelligence, artificial intelligence, but not inappropriate time. Okay, so Ms. Emanuele will talk about biocompatible nanostructure system incorporating natural products in bioactive molecules derived from food. So, okay, is it okay, Emanuele? So she's from the food technology area, and she's our last speaker today. Não tá indo pro... Ah, tá legal. Aham. Okay. So from 10:40, we are going to, inside university, we are going to see the plant nutrition laboratory. We can now see the aeroponics. We don't have plants now, but the structure are there. And we are planning to go to the laboratory of the biocontrol. And also there is another laboratory about gene research. And if we have time, we're going to see Dr. Nagy laboratories. But Professor Nagy, I don't know if, okay, we will manage to go to your laboratory too, okay? At the end. Okay. And as I said, there's coffee, there's some pão de queijo out there, and feel free to take. Oh, coffee is finished. Okay. Very high.
Sim. Eu dava aula de inglês na física na minha cidade. Eu ainda não conectei o professor, senão meu computador ele trava.
Não quero mandar o vídeo no computador?
Já consegui aqui. Só tô...
Você tem passador?
Não.
Vocês vão
Okay. Não precisa? É? Nem o point? Tá bom. É na parte vermelha. Okay, so shall we start? Please, Mrs. Emanuele, we have to leave here 10:40, okay?
So, good morning, everyone. Sorry for the delay because my computer is a bit low. So, my name is Emanuele Firmino. I am a graduate student at the Nanosciences and Biosciences program under the supervision of Professor and Dr. Najma Issar Khalil. I work in the equipe of the Applied Non-Structured Systems Laboratory. So when we talk about nanotechnology, we has the concept that has been widely studied in different fields. So because of that, our laboratory aims in green synthesis techniques who considerate fast, simple, and scalable developments consistent in nanocompounds with organic or inorganic compounds like zein, ketosene, silver nanoparticles, and gold nanoparticles. Various of our studios, we need trials to characterize these nanoparticles using components like size, zeta potential, encapsulation efficiency, and stability to has a reliable result in a physical-chemical analysis of these components. With these components and parameters, we enter in the assays instruments, which we conduct such assays with antioxidant, cytotoxicity, assays antifungic, antibacterial, and we have like other assays, we use animal models to check diabetic parameters, pharmacokinetics, and toxicity and antitumoral effects of these natural compounds. So applications of our previous research group, we have as a vast study on curcumin extracted from curcuma longa, where in this study, when we administrated in rats in its free form, it has a low bioavailability in the plasma of these rats, reducing its effects. And when we nanoencapsulate its compound, we can enhance its beneficial effects by viabilizing more bioavailability of curcumin in plasma of these rats. Other natural compounds we found mainly in red fruit, colored fruits is the gallic acid. When we encapsulate it, we observe that when we have a reduced cytotoxicity in cells, and we enhance the antioxidant activity because of the controlled and cumulative release on the body. Plus, we have another curcumin model with to check the anti-tumor activity on murine melanoma model. So we tested his effective treatment on tumor cells, and we checked that the volume of growth of cells with tumoral effects has reduced significantly when we administered the curcumin nanocapsules. When we talk about other components, we can say in these actual studies, we have the resveratrol that's found commonly in grapes. And when encapsulated, we can reduce the tyrosine hydroxylase. It's a protein level that when it's low in our body, it's expressed by MPTP rats and contribute to modulate and treat a chronic disease treatment such Parkinson. And when we talk new applications of natural compounds and extracts in the field application, we have the metal or organic nanoparticles, nanoemulsions, nanocrystals, with among other materials and components that can serve with as an important tool for improving fuel production, enhancing the efficiency of resources for pest control, and we can reduce costs and minimize the diverse effects when in the field plantation. So our laboratory counts on with international collaborations and research grants supported by CNPq and CAPES from Brazil in partnership with institutions from Canada, United States, Belgium, Portugal, and Germany. Also, if possible, the laboratory is always interested and willing to collaborate with new institutions, such as Japan and the university, participating in future projects and or extension researches. We not only do institution researches, we collaborate with projects in industries with pharmaceutical and cosmetics industries. Sorry about the nerves, but I would like to thank about the attention and the patience. I hope that was understandable for our guests, and thank you.
Okay, thank you, Ms. Manueli. You are student, grad student, undergrad? Master or PhD? Master student? Okay, maybe you can also do some research in Japan also, right? Okay. Well, we have 5 minutes. If you have some questions, some comments, please, Professor Watanabe.
Hay una diapositiva respecto a respirador de uva. Entonces, maracuyá también tiene, semente de maracuyá tiene respirador. Y hay alguna pesquisa respecto a la semente de maracuyá.
Potassium fluoride is interesting because it has anxiolytic activity. It's interesting.
Okay.
But nanoparticle, I don't know now about the potassium fluoride. It's interesting because the anxiolytic activity this word. Yeah. Not necessarily. Just the juice.
Because usually seeds are like a waste, and Japan import a lot of the passion fruits products. And one company looked at how they can make better use of seeds, and they looked at the presence of resveratrol. And they are looking at particularly use of seed of maracuya for product development. You have a lot of the maracuya. Obrigado.
Okay, someone else? Other questions? Well, today we were talking from professors from the Department of Agriculture and also for pharmacy. And we hope in the future we could do some joint research, and also that our students can have the opportunity to go to Japan. It's an amazing place. It's a very different because of the culture, but it's amazing place to visit. Okay, so I don't know, should we take a photo now here? Okay, from this section, and after that we are going to go to visit our laboratory. We're going first to the soil nutrition lab, and after that we are going to see the aeroponics, and after that we are going to the other two laboratories. Okay, bio protection and also the Professor Paulus. Thank you.